or Good day po. My name is Diana Lea Mendoza. I will be discussing the second series of immunology and serology review questions for ASCP board review. Okay? So, I have 65 questions, and these are helpful, very helpful questions that are gathered from BOC, from Lab CE, from HAR, Shula, and from other immunology and serology textbooks. Very helpful po itong mga questions ito sa akin. Hopefully sa inyo rin po, maging helpful din po. Okay? So, first question. Number 1, skin, lactic acid secretions, stomach acidity, and the motion of cilia represent which type of immunity? Choices are natural for letter A, B, acquired, C, adaptive, or letter D, passive. For this question, the answer is natural immunity. So the answer is natural immunity. So remember that skin and mucous membranes, along with other functions such as phagocytosis, complement, acute phase proteins, and others are all part of natural, or also known as your innate immunity. Okay? So this gives protection that is non-specific. Okay? It gives non-specific and is readily available, okay, from the patient or from the individual's body build. So, ito po yung kasamahan ng tears or luha, saliva, earwax, okay, that can be an external examples of external, okay, natural or innate immune, okay, system. Meron din tayong second line of defense mechanism under natural immunity, which includes our phagocytes, okay, that functions in phagocytosis, okay, and our complement molecules, okay, and our acute phase proteins. Okay, so mamaya meron po akong pin-prepare na mga discussions, okay, ulitin natin. Okay, what is acquired immunity? Acquired immunity is also known as an adaptive immunity. So these are defined, okay, by the presence, okay, of lymphocytes, either T or B cells, okay, and T and B cells, okay, and are effector cells that directly destroy a specific antigen. Okay? And this type of immunity is involved in the recognition of this foreign antigen, okay, and a specific humoral or antibody or cell-mediated response. Okay, so they are both, okay, termed as acquired, okay, or adaptive or specific immunity. When we say naman po passive, this is the term used, okay, or the term that we use when antibody is formed in one individual, okay, that is given to another individual who is at risk, okay, of infection. Okay? So we have two types of immunity. First, we have innate immunity, okay, or we call as natural or non-specific. So again, sabi nga natin, it is the ability of an individual to resist infection by means of normally present body function. So these are the examples. Kanina, sabi natin, we have the physical, okay, and chemical barriers. These are our external defense mechanism. Okay, includes intact skin, skin with lactic acid, keratin, tissue structure. Di po ba yung skin natin, this is made of stratified squamous keratinizing epithelial tissue. So, patong-patong yung cells, okay, preventing the abrasion, preventing the passage of pathogens, no? So, we have also mucosal membrane surface, which includes, for example, in our respiratory tract, we have cilia. Okay, so kapag meron pumasok dyan na foreign molecule or foreign objects, okay, so nagde-trigger yung cilia natin na i-flush away, okay, yung mucus na pinroduce sa ating respiratory tract palabas ng ating, okay, ng ating respiratory tract. Okay, what else? We have secretions, okay, this includes your sweat, mucus, earwax, oil, tears, and saliva. Okay? So we have this chemical barriers. Okay? These are found in the tissues. Okay? These are the complement, interferons, and lysozymes. Okay? And then yung kanina sabi nga natin, okay, we have the actions of phagocytes, okay, which is needed in the phagocytosis, okay, which will later on trigger also, okay, inflammations. And we have complement system, okay, and then acute phase, okay, reactants. Okay? Second, we have the adaptive immunity or specific or acquired immunity. So, this is acquired, okay, only after a specific exposure, okay, only after a specific exposure or infection is encountered, okay, and responds specifically, okay, specifically to that infection. So, what are the two responses under adaptive immunity? We have one, we have humoral mediated and we have cell mediated. Humoral mediated, its mechanism is antibody mediated, okay, mediated by by B lymphocytes, by B is to produce over serum. Okay, so its purpose is to provide primary defense against bacterial infection. Okay, we have the cell-mediated immunity. Okay, this involves T lymphocytes or T cells. Okay, the mode of action: direct cell-to-cell contact of soluble products secreted by the cells. Okay, its main function is to provide specific defense against viral, fungal infections, okay, intracellular organisms, tumor antigens, and graft rejection. Okay? So, for number 1, the answer po is natural immunity. Number 2, the inhibitory receptor expressed by NK cells, okay, bind to which of the following in order to prevent killing of normal cells? Letter A, complement receptors. Letter B, MHC class 1. Letter C, immunoglobulins. Or letter D, toll-like receptors. Okay, so again, di ba na sa ating previous discussion and in our lecture notes, okay, natural killer cells, they are lymphocytes. They are a subclass of leukocytes, okay, in our circulation, in the blood circulation, that recognize virally, okay, recognize virally infected and neoplastic or malignant cells. Okay? So, they recognize these cells, these virally infected and neoplastic cells, as, okay, non-self. Okay? So, these natural killer cells, they are considered as non-B, non-T lymphocytes, okay, that do not require a specific antigen activation to detect this this particular cells, the virally infected, and neoplastic cells. Okay, so NK cells are very important in destroying this types of cells and limiting the progression of malignancy or cancer. Okay, in general, NK cells do not require antigen-specific recognition to kill target cells, but they do, however, recognize recognize MHC class 1 molecules, our MHC class 1 molecules, as self. So therefore, this natural killer cells, once they recognize MHC class 1 molecules through their surface receptors, they deliver inhibitory rather than activating signal. So, hindi niya papatayin. Okay? So, for this, number 2, the inhibitor expressed by the NK cells bind to which of the following order to prevent killing normal cells? The answer is letter B. Okay, letter B. Okay, number 3. B lymphocytes and T lymphocytes are derived from letter A, hematopoietic stem cells, B, macrophage or monocytes, C, mucosa-associated lymphoid tissue or MALT, letter D, granulocytes. So for the answer in number 3 is letter A. Okay, why? All lymphocytes originate and derive from hematopoietic cells or stem cells via hematopoiesis. So the stem cells are will eventually mature into common lymphoid progenitor cells. Okay, so the stem cells will later on be maturing to common lymphoid progenitor cells. Okay, these progenitor cells will then differentiate into distinct types of lymphocytes. Okay, so pwede siyang T lymphocytes, pwede siyang B lymphocytes, and further mature into a more specific type of those cells. So B cells mature into B lymphocytes in the bone marrow. Okay, the B cells mature, so we have your B cells. So they mature in the bone marrow, while T cells, mature in the thymus. Okay, in the thymus. So again po, for maturation of our cell types, the B cells will mature in the bone marrow, while your T cells will mature in the thymus. Okay, so for number 3, B and T cells are derived from hematopoietic stem cells. Letter A. Letter A. Number 4. Characteristics of type 1 cryoglobulins include the following characteristics. Letter A contains 2 classes of immunoglobulins, at least one which is monoclonal. Letter B, mix, no monoclonal protein found. C, monoclonal IgA, IgG, or IgM. Or letter D, contains 5 classes of immunoglobulins. So, let's define first what is a cryoglobulin. So, cryoglobulins are circulating proteins, specifically immunoglobulins. Okay, so these are the IgG, IgM, IgA, or light chains. Okay, so they clump together. Okay, that clump together, forming precipitates. Okay, when they are exposed to cold and dissolve when warm. Okay, so again, cryoglobulins, these are circulating proteins. Okay, it can be our immunoglobulins, okay, that clump together forming precipitates, okay, when exposed to cold and later dissolve when exposed to warm. So we have types of um, um, cryoglobulins. Question, bakit ba kapag nag-precipitate ang cryoglobulin, what will happen or what can it cause to the body? So precipitated cryoglobulins, they can slow the flow of blood, okay, and this can block small blood vessels. Okay, so again, precipitated cryoglobulins can block, okay, and slow the flow of blood in the blood vessel. So presence of large amounts of cryoglobulins, we call this as cryoglobulinemia. Okay, so cryoglobulinemia, which can cause symptoms such as bruising, rashes, joint pain, weakness, okay, and pain, paleness, okay, numbness, okay, tingling and coldness of the fingers and toes, okay, especially when when these individuals are exposed to cold. Okay, so we have different types of cryoglobulins. Type 1, cryoglobulin, type 1, okay, type 1. So this cryoglobulins these are your monoclonal IgA, excuse me, IgG and IgM. While your type 2 cryoprecipitate is mixed. Okay, this is mixed. Okay, containing two classes of immunoglobulins. Okay, at least one, okay, of which is monoclonal. Okay, at least one of which is monoclonal. Okay, so we have your type 3. Your type 3, okay, cryoprecipitate. Okay, this is mixed. And no monoclonal protein is demonstrated dito with one that is monoclonal for type 2 clonal, okay, while yung type 3, this is also mixed, okay, containing immunoglobulins which are considered as monoclonal or no monoclonal protein is demonstrated. So again, type 1 cryoprecipitate is monoclonal such as your IgG, IgA, or IgM. Type 2, these are mixed type of cryoprecipitate which contains two classes of immunoglobulins. Okay, at least one of which is monoclonal. That is type 1. Type 3, okay, this is mixed. Okay, mixed and no monoclonal, okay, and no monoclonal protein is present. Clonal protein is present or demonstrated. Okay, so for this question, the answer is letter C. Okay, letter C. Okay, monoclonal IgA, IgG, or IgM. This is the type 1 cryoglobulins. Okay, we have your type 1 cryoglobulins. Okay, number 5. Which of the following types of molecules is most likely to be antigenic or we call as immunogenic? So we have your nucleic acids, letter A, B, carbohydrates, C, lipids, or letter D, proteins. So number 5, the answer is letter D, proteins. So given that proteins have high molecular weight, okay, property, they also show structural complexity. Okay, so proteins are excellent, okay, antigens. They are more antigenic compared to the other 3 listed in the list. Okay, so lipids are considered as inferior antigens because of their relative simplicity and their lack of structural stability. But once lipids are bound with proteins and carbohydrates or polysaccharides, they can function, okay, as good antigens. Okay, they can function as good antigens. Carbohydrates, on the other hand, okay, they are less antigenic. Okay, they are because they are considerably too small to function as antigens. Nucleic acids are not generally antigenic. Okay, nucleic acids are not, okay, generally antigenic. They are poor antigens, okay, because of relative simplicity, because of their molecular flexibility and rapid degradability. Okay, madaling masira ang nucleic acid. So for number 5, the answer is letter D, proteins. Number 6, which complement component is found in both the classical and alternative pathway? We have letter A, C1, B, C4, C, factor B, and we have letter D, C3. So for number 6, the answer is C3. Okay, the answer is C3. Okay, so C3 is present in the plasma in the largest quantities. Okay, so fixation of C3 is the major quantitative reaction of the complement cascades. So we have 3 pathways. We have your complement system, your classic, and alternative. We have your 3 pathways of the complement system. We have your classic, we have the alternative, and mannose binding lectins. They are initiated in various ways. We have for classical pathway, this is initiated by immune complexes, by apoptotic cells, by certain viruses, and gram-negative bacteria, and C-reactive protein that is bound to a ligand. Okay, so CRP, okay, that is bound to a ligand. So this is the initiator of the classical pathway. For the alternative pathway, we have various bacteria, fungi, viruses, or tumor cells. Okay. We have also the mannose binding lectin. Your MBL or your mannose binding lectin This is initiated by microbes with terminal mannose groups. So these three complement pathways converge at the point of cleavage of C3 and C3b, okay, the central event of common final pathway, which in turn that will lead to the activation of the lytic complement, okay, sequence, okay, C5 to C9, and cell destruction. So the protein C3 is needed in the production of your C3b, which will bind to the surface of microbe where it function as an opsonine, okay, and a component of C3 and C5 convertases. C3a, okay, is a good stimulator of inflammation. Okay, so we have C1. C1 is active in the initial phase of complement activation. The classic complement pathway is initiated by the binding of the C1 complex consisting your C1q, C1r, and C1s to antibodies bound to an antigen on the surface of a bacterial cell wall. So again, C1, it is a molecule, okay, that initiates the classical pathway. C1q binds to the Fc portion of the antibody. C1r and C1s are proteases that lead to the C4 and C2 activation. We have your C4. The cleaving of C4 protein will lead to the production of C4b. Okay, C4b covalently bind to the surface of microbe or cell where the antibody is bound and the complement is activated. C4b binds to the C2, okay, for cleavage by C1s. Okay, C4a, okay, is an acute phase reactant, okay, and it is a good stimulator of inflammation. We have your factor B. Okay, factor B, okay, this is found in the alternative complement pathway. Okay, the factor B component is consumed by the activation of the alternative component pathway. So factor B, okay, in the form of BB, okay, this is a serine protease and the active enzyme of C3 and C5 convertases. A deficiency of factor B is associated with hemolytic uremic anemia or the atypical hemolytic uremic syndrome. Number 6, the answer is letter D. So, we have number 7. 7, ability is best described by which of the following statements? Letter A, the strength with which red cells agglutinate. B, the strength with which multivalent antigens and antibodies bind. C, the strength with which univalent antigens and antibodies bind. Or letter D, the speed with which an antigen-antibody reaction occurs. So for number 7, the answer is letter B. Okay, the strength with which multivalent antigens and antibodies bind. So the term avidity, okay, it refers to the total strength of the attraction between the antibody and a multivalent antigen. So the reaction between an IgM molecule, okay, your IgM molecule which is a pentamer containing 10 antigen binding site, okay, and a multivalent antigen, okay, is therefore, okay, considered a stronger, okay, antibody molecule compared to the IgG antibody. IgG antibody, okay, contains 2 antigen binding sites, okay. So this will give, okay, your IgM, okay, your IgM a capacity, okay, more capacity of binding, okay, the antigens. So, the term affinity, on the other hand, it refers to the strength of attraction, okay, between a single antigenic, okay, determinant and a corresponding antigen binding site. For number 7, the answer is letter B. Number 8, most antibodies present in cord blood are of blank origin. We have A, fetal, B, maternal, we have C, paternal, or letter D, maternal and paternal. So the answer for number 8 is letter B, maternal. So infants who are considered as less than 6 months old usually do not produce alloantibodies. So newborns may have passive antibodies, okay, from maternal origin. Okay, from maternal origin, galing kay mommy. Okay, these antibodies, okay, cross the placenta. Okay, they cross the placenta, but they do not have, okay, passive antibodies coming from the father or from a paternal origin. This is from the mother. So for number 8, the answer is letter B, maternal origin. Number 9. Which of the following types of lymphocytes express CD4? We have A, only T helper cells, B, only T suppressor or cytotoxic cells, letter C, all T cells, or letter D, B cells. So the answer for 9 is A, only T helper cells. So T helper cells express CD4. Okay? So T helper cells express CD4. Okay? Neither T suppressor cells nor B cells express CD4. So CD3 is expressed, okay, on the other hand, we have CD3. Okay, CD3 is expressed by all, okay, CD3 is expressed, okay, by all mature T cell. All mature T cell, including the T helper, okay, and T suppressor, okay, T cell. Okay. So CD3 is expressed by all mature T cell, including the T helper and T suppressor cells. So number 9, the answer is letter A, CD4. Number 10. Which one of the following statements about endocytosis is incorrect? Number 10. Which one of the following statements about endocytosis is incorrect? Letter A, phagocytosis is a form of endocytosis. B, in endocytosis, the membrane invaginates and isolates a foreign particle in a vesicle. C, endocytosis is mechanism for viruses to enter cells. Or letter D, endocytosis is the process by which phagosomes expel the macromolecules from the inside. So A, B, and C correctly describes endocytosis except letter D. So the exception is letter D. So endocytosis is the reverse process of exocytosis, which is described here. So exocytosis is process of expelling a foreign material. Okay, so expelling, okay, a foreign material. This is described in letter D. So phagocytosis is a form of an endocytosis. This important body defense mechanism is a process where the specialized cells, which are your phagocytes, engulf and destroy foreign particles such as microorganisms or damaged cells. Okay, we have your macrophages and segmenters or segmented neutrophils that are considered as the most important Okay, most important, phagocytic cells. So remember, in endocytosis, the cell membrane invaginates and forms vesicles to isolate foreign particles. And endocytosis is the mechanism for viruses to enter cells. For number 10, the answer, exception po ang letter D. Okay, exception po or incorrect po ang letter D.